Assalamualaikum friends. Today I'm going to present basic terms of genetics. Friends, these terms are really very important for understanding before understanding any processes related to genetics. I have already downloaded a video in which few of the terms are elaborated. If you want to see that video, then please go into the description box where I have given the link of that video. So let's start the session. Friends, I try to avoid copying dictionary definitions over here because my intentions are just to make you understand what these definitions are. The first definition is clone. Clone is nothing but creating copies of organisms. It might be cells or it might be DNA fragments. Basically, scientists use asexual reproduction method for the production of clones. You can say they take somatic cells for clone production. As you can see in this picture, all of the shapes are of same type or copies of one another. Now strain. Friends, let me give you example. I have a certain sort of sample and now I have to culture from that sample. When after culturing I found influenza viruses over there, but I have when do further study then I just get then some of the species possess different characteristics that they can produce flu in the living organisms. Why these some of the species are different from the rest of the species? Because they possess different isoforms of the surface proteins. Just because of this genetic variant, they are able to produce flu in the living organisms, while otherwise all of the genetic makeup they possess is same as the rest of the species. So these special species are termed as strain. Friends, you can simply say a strain is a subtype of a microorganism, or you can say the strain is a genetic variant. Genome is nothing but all the genes present in a cell or virus is called genome. I think this is the simplest definition of this genome. After genome, you must know haploid and diploid terms. You can see in this picture at one side chromosomes are present with its pair, or you can say with its copies. But another side you can see chromosomes are alone, they do not contain or possess any type of copy. So in this type where chromosome possesses copy, it is diploid, and when the chromosome do not possess copy, you can say one copy of each chromosome is present in a cell, then it is called haploid. Friends, normally bacteria have one set of genes, so that they are haploid, or you can say 1n, and eukaryotic microorganisms have two sets of genes, so that's why they are called diploid or 2n. Now, the terms of genotype and phenotype. You must know the difference between these two terms. Let's understand these terms with this picture. Friends, capital letter always represents dominant character of a gene and small letter represents recessive character of a gene. In this picture, capital B shows purple allele while the small b shows white allele. When a cell contains purple alleles or dominant purple alleles, then this is the genotype of a cell. When this character appears physically and you observe or you can see this character in that flower that is purple, this is called phenotype. In another picture you can see that a cell contains dominant purple color and a recessive white allele. Definitely purple color represents because it is a dominant gene, so the color appears purple and physically appearance of this color is called phenotype, while the presence of these two genes in a cell is called its genotype. In the third picture, two of the recessive white alleles are present, and so they appears as a physical character of the white flower, and this is called phenotype. When the presence of the genes within a cell is called genotype, and the appearance of that particular genes within a living organism is called phenotype. Genotype is the genetic characteristic of an organism, while the phenotype is the physical characteristic of the organism. The most simplest definition: when a human possesses blue eyes, and the appearance of that blue eyes is phenotype. When it's lacking brown eyes, it means it's lacking that particular type of gene, or that is its genotype. Friends, three terms in which we always confuse, that's the genetic engineering, recombinant DNA technology, and that's the biotechnology. Let me explain. Firstly, genetic engineering. Deliberately, intentionally making changes in the genetic information, or you can say directly in the nucleic acid genome, is called genetic engineering, as you can see in this picture for more clarification. Recombinant DNA technology, friends, for the accomplishment of that methods, for the accomplishment of genetic engineering, certain methods are used, and these methods and the use of these methods, you can say collection of these methods, is called recombinant DNA technology. You can see in this picture that the recombinant DNA technology at the certain method has been applied in order to do this phenomena, or in order to make changes in the genetic information, as one of the cell is a human cell and another cell is of E. coli bacteria, and they are intentionally making changes in their both the genomes by following a technology, or you can say method, and that is called the recombinant DNA technology. Friends, now what is biotechnology? Basically, recombinant DNA technology is an essential part of the biotechnology. Biotechnology is a very broad area of the biology in which living organisms, we can say living systems, were used for making the usable products, or develop the usable products which are beneficial for human beings. That is called the biotechnology. Guys, hope you understand all of these basic terms very clearly. I tried my best in order to elaborate it simply and try to make you understand. If you have still any query, then please comment in the comment box. I will resolve your issue and we'll meet in my next video. Allah Hafiz.